Alright, today we're standing outside of our radiotracer lab. This is where we deal with all of our radioactive materials for labeling phytoplankton or metal binding ligands in the ocean, for doing experiments at really low concentrations where we can get the answers quite rapidly. So we start the experiment in the morning, and by the afternoon we have all of our answers. Come on in. So in this lab, you'll notice on the floor, I'm taking off my shoes because we have funky moon boots in this lab. And the reason for that is, while we are very careful with radioactive materials, we would hate to have a situation where our street shoes contaminate the hallway with any kind of contamination from isotope spills. And we also do work, we try to achieve trace metal clean conditions in this lab. So again, bringing street shoes in with all the mud and snow and slush is not a good idea. So we have these moon boots in 3 assorted colors. Okay, I get the black ones. We haven't seen anybody wear the pink ones yet, but I suspect on the weekends they might. Okay, so anyway, beyond this red line, we try to treat the place as generally trace metal free. It's not entirely true. Where that becomes more true, and everything is relative, is inside this bubble that we constructed. Okay, so in the rad lab, of course we're always wearing coats and jackets and goggles and gloves. And what we see here on the floor, this is our shipment of lysates ready to go to Antarctica. So these are lysates of diatoms and Phaeocystis globosa, which is related to Phaeocystis antarctica. What we've done is we've labeled cells with either iron-59 or C14 as the radioisotopes. We've also done a C13 treatment so we could follow the incorporation of regenerated carbon into the DNA of heterotrophic bacteria. Okay, so come on in here. So we might have to, so this room here is filtered through a 0.2 micron filter, so the whole space is under positive pressure. We keep the fan running when we're not here. But when we come in, we actually turn up the volume, or turn up the speed on the fan. This is a handiwork of my technician and I. And this way we really crank out the positive pressure so that any dust or any foreign particles don't actually contaminate our samples. So working with radioactive materials requires you to be double gloved, and most importantly, it requires that you have university certification, which requires a training course and then an annual refresher online. And equally important is the training that I give to all my students and technicians on the proper use of radioisotopes in the lab. You'll notice over here, if you are working in this lab, there's basically a document that describes how we treat the radioactive materials in the lab, because very often we will be using, for example, polycarbonate tubes we use more than once. So after they've had iron-59 in them, we have to use them again, clean them, and make sure that they're trace metal free, radioisotope free, so they can move on to the next experiment. Okay. So when you're working with iron-59, you have to wear a ring badge and a whole body badge, which basically is a way to monitor your occupational exposure to radioisotopes. So inside here we have our trace metal clean reagents that we use when we're sampling phytoplankton, sampling the lysates. So we're using the radio label to trace the fate of carbon and iron in Antarctica, in the Antarctic assemblages. We offer them some of this radio label goo. But we also want to be trace metal clean about it. We want to make sure that we know the amount of cold or non-radioactive iron in the sample so we can properly calculate the total iron incorporation rate into the phytoplankton. Okay, what I'm holding here is actually not radioactive. This is one of our C13 lysates that we're dealing with. You can see this is about 1 liter of culture, which we filtered out and resuspended into about 4 or 5 mils of Helix phosphate buffer solution. Get that